Okay, let's unpack this. We're opening the file on a molecule that has, well, it's quietly driven the last decade of scientific revolution, yet for so long, it was basically a footnote.
It really was.
Welcome to the deep dive, where we take these vast, complex topics and, try to extract the essential knowledge just for you.
Today we're going deep on RNA, ribonucleic acid. And it's so often been cast as, DNA's dutiful assistant.
The biochemical backup singer. I love that phrase.
Exactly, just carrying instructions. But the sources we've looked at, they show that RNA hasn't just stepped into the spotlight, it's rewriting the entire play.
That's a perfect way to put it.
Okay.
We are living in what is, well, indisputably the age of RNA, and the proof isn't just theoretical, it's right there in the numbers.
It really is. I mean, think about this. Since the year 2000 alone, breakthroughs directly tied to RNA have won 11 Nobel Prizes.
11. That concentration of recognition in just over 2 decades, it's just staggering. It tells you where the revolution is happening.
And that focus is fueling an absolute explosion of practical application. The number of peer-reviewed articles, the number of patents coming out of RNA research, it has quadrupled in that same time frame.
That's an exponential leap. It's not just steady progress.
Not at all. It speaks to a fundamental shift in our understanding.
And crucially, this isn't just staying in the lab. We're seeing real, tangible medical impact. Right now, there are over 400 RNA-based drugs in development.
400. And if you want a really clear signal, look at the money.
Right, market confidence.
In 2022 alone, private equity funds poured over $1 billion into biotech startups focused specifically on RNA therapeutics. That is a massive bet on a molecule that, a lot of people used to just dismiss.
So to really appreciate this, this transformation, we have to go back to basics. We have to draw a really clear line between RNA and its, let's say, more famous relative, DNA.
Right. DNA is, for all intents and purposes, a passive library. It's the master blueprint, the single source of truth locked away for safety.
I love the analogy the sources use here, that DNA has just one trick. It stores information perfectly. It's like hieroglyphics carved in a tomb or the grooves on a vinyl record. It holds the music, but doesn't play it.
And because that job is so critical, in higher organisms, it's locked away safely inside the cell nucleus. It stays passive. It stays stable.
RNA, on the other hand, RNA is all about action. Its defining feature is its incredible versatility.
Yes, it can store information. I mean, plenty of viruses use RNA as their entire genome, but that's just scratching the surface. RNA is a structural engineer, it's an enzyme, it's a communications expert, all in one.
And the reason it can do all of this, it comes down to its shape, right? It's this tiny little thing, a nanometer wide, and it folds into these amazing, complex, almost origami-like structures.
And that three-dimensional form gives it the power to act. It's not just a string of letters anymore.
It's a machine.
It's a machine. It can be an enzyme, a catalyst to speed up chemical reactions. It's a core part of the machinery that builds proteins. It powers telomerase, that enzyme linked to aging in stem cells. And, of course, it's the guide molecule, the brains behind the whole CRISPR gene editing operation.
So that's our mission for this deep dive. We're going to trace this incredible scientific journey. We're taking RNA from this overlooked, unstable molecule
all the way to its current status as life's great catalyst.
We'll look at its role in coding, catalysis, aging, disease, and even the very origin of life itself.
It's quite a story.
It is. So let's start at the beginning. Part 1, RNA's first job, the messenger and the code.
The story really kicks off in the 1950s, right after Watson and Crick figured out the double helix. DNA was the star of the show, but a few key people were already seeing a problem, a fundamental paradox.
And one of those people was, well, not a biologist at all. He was a physicist named George Gamow.
An incredible character, a giant 6'6" Russian physicist, one of the fathers of the Big Bang Theory. But he was also famous for his sense of humor, just a complete refusal to take himself too seriously.
Is he the one who put his friend's name on a paper just for a pun?
That's him. He added Hans Bethe's name to a paper he wrote with Ralph Elfer, just so the author list would be Alpha, Beth, Gamma. Like the first 3 letters of the Greek alphabet.
Alpha, beta, gamma. That's fantastic.
Pure genius, but also a total prankster. And this was the mind that turned to cracking the genetic code. Gamow saw that the challenge wasn't just what the code was, but how it got from the DNA to the protein factory.
So he put together this secret society almost, the RNA tie club.
A very exclusive club, a small fraternity of the world's top code breakers, and he sent each of them a woolen tie embroidered with an RNA strand.
But wait, why RNA? Why not a DNA tie? DNA was the big discovery.
Ah, well, that's because Jim Watson had convinced him. Watson pointed out the core architectural problem in all higher life forms. It's a spatial problem.
Okay, lay it out for.
If DNA is the master blueprint and it's locked securely inside the nucleus, how does that information get out to the protein-making factories, the ribosomes? Because the ribosomes are out in the main body of the cell, the cytoplasm.
Right. You need a courier. Something has to carry the message across the border from the nucleus out into the cell.
Precisely. You need a temporary, disposable message. And RNA was the perfect candidate. They already knew it was abundant in both places, the nucleus and the cytoplasm.
And chemically, it's almost identical to DNA, but with this one key difference.
One extra oxygen atom. RNA is ribonucleic acid. DNA is deoxyribonucleic acid. That little oxygen makes RNA much less stable. It breaks down more easily.
Which for a long time was seen as a flaw, right? There's another reason everyone focused on proteins.
Right.
Proteins were stable, complex, they did things. DNA and RNA were seen as, well, kind of simple and boring.
They were seen as just passive information storage. But once the idea of a messenger took hold, the search for messenger RNA, or mRNA, began in earnest, and the first results were deeply confusing.
This is a great part of the story. They were looking in the right place, but they kept finding the wrong kind of RNA.
Exactly. They had analyzed the RNA in the cytoplasm, and the vast majority of it was what they called rock stable. It just stuck around. And worse, its composition was always the same. Meaning, whether the cell was making, say, insulin or hemoglobin, this stable RNA always had the same proportions of its 4 bases, A, G, C, and U.
That makes no sense if it's a message. That's the key stumbling block. It's like saying the sheet music for Beethoven's Ninth has the exact same notes in the same proportions as Lady Gaga's Bad Romance.
Right. You'd have to conclude the sheet music isn't carrying the unique instructions at all. So this dogma held until a breakthrough in 1960. It took a few brilliant minds connecting the dots from different experiments. Francois Jacob, Francis Crick, and Sidney Brenner.
And they remembered this weird old data from a couple of researchers at Oak Ridge, Vulcan, and Astrakhan.
Yes. Vulcan and Astrakhan had been studying E. coli bacteria infected with a virus, a phage called T2, and they saw something strange happen right after the virus attacked.
What was it?
A huge, rapid surge of new RNA molecules. But these were different, they were smaller, and they were short-lived. They appeared and then disappeared.
The transient messenger.
And crucially, the base composition of this new, unstable RNA matched the viral DNA, not the bacteria's DNA. Jacob, Crick, and Brenner realized this was it. This was the elusive mRNA. The stable RNA everyone kept finding was just the ribosome itself, the rRNA, the factory machinery.
So the real message was like a snapshot. It delivers its information and then vanishes.
Perfect analogy. The ribosome is the turntable, the permanent machine. The mRNA is the vinyl record, the specific instructions for one song. And the protein is the music. The mRNA is temporary. The ribosome is constant.
Okay, so they found the messenger. Now they have to figure out how it's read. And this goes back to Gamo's idea about a triplet code.
It does, and Crick's colleagues confirmed it with some really clever genetic experiments. They looked at what are called frame shift mutations.
Right, so if the code is read in groups of 3 letters, like words, what happens if you add an extra letter?
If you add 1 or 2 extra bases, it scrambles the entire message from that point on. The reading frame is shifted, and you get gibberish. That's what causes horrible diseases like cystic fibrosis.
But what if you add 3 letters?
If you add 3, you're just adding 1 extra nonsensical word. The rest of the sentence after that word is back in the correct reading frame. And they found that proteins made this way were, mostly functional. It was definitive proof. Codons are triplets.
The final step was to actually translate one of these words, and that fell to Marshall Nirenberg.
Nirenberg and his postdoc, Heinrich Mathai, in 1961. And they did it in the simplest way possible. They didn't even use a real gene.
No, they just made a fake one, right?
Yeah.
An artificial RNA strand made of nothing but 1 letter, uracil. They called it poly-U.
Exactly. They put this poly-U string into their protein-making machinery in a test tube, and the protein that came out was made of only 1 amino acid, phenylalanine.
Wow, so U equals phenylalanine. That was the first word in the genetic dictionary.
It was the Rosetta Stone. It opened the floodgates, and soon they had deciphered all 64 possible codons.
And there was one last piece of this information puzzle, the adapter molecule.
Found by Paul Zemechnik. He was using radioactive amino acids to trace the process, and he noticed something odd. The amino acids didn't just float over to the ribosome. First, they became attached to these other very small RNA molecules,
These became the transfer RNAs, the tRNAs.
the tRNAs. The physical adapters that read the codon on the mRNA and transfer the correct amino acid to the growing protein chain.
So by the mid-60s, the picture is pretty clear. You have mRNA the message, RNA the factory, and tRNA the delivery truck. RNA was the whole logistical network.
Yes, but it was still seen as just that logistics, the grunt work for the all-important DNA. No one had yet grasped its secret powers.
Which brings us to part 2, RNA's secret powers, splicing and ribozymes.
The idea of RNA as just a simple assistant was completely shattered in the late 1970s. Two huge discoveries came along that showed RNA had deep regulatory and even catalytic power.
The first bombshell dropped at the Cold Spring Harbor Symposium in 1977, and it was the discovery of RNA splicing.
Right. Up to that point, biology had this guiding principle, the monad principle. Anything found to be true of E. coli must also be true for elephant.
Meaning biology is universal. The basic rules should be the same everywhere. And in bacteria like E. coli, genes are simple and continuous. The DNA is copied straight into mRNA, which is then translated. Clean.
Everyone just assumed human genes work the same way. But when Phil Sharp and Rich Roberts looked at how mRNA was made in human cells, they hit a paradox.
The size problem.
The size problem. The initial RNA copy made in the nucleus was often 10 times larger than the final mRNA that got shipped out to the cytoplasm.
So where was all that extra stuff coming from, and where did it go?
They discovered that our genes are not continuous. They're fragmented. The DNA itself contains long stretches of, well, non-coding sequences that were named introns.
So the cell copies the whole thing, introns and all, into this massive precursor RNA.
And then before that message can be used, all those introns have to be precisely cut out, and the important coding bits, the exons, have to be stitched or spliced back together perfectly.
It seems like such an inefficient, bizarre step. Like writing a chapter for a book, but deliberately scattering paragraphs of gibberish throughout that you have to manually delete before you can print it.
It's a great analogy. The sources describe it like a sailor finding a frayed section in a rope, cutting out the bad part, and splicing the two good ends back together so seamlessly you can't even tell it was cut.
This discovery led directly to what became known as the human genome paradox.
Ah, the shock was immense. When the full human genome was sequenced in 2003, we all held our breath, and the number came back. Around 24,000 protein coding genes.
Which, not a lot.
It's about the same as a tiny little worm. It's only 4 times as many as a single-celled yeast. The question was immediate. How can you build something as complex as a human being, with our brains, our immune systems, from such a modest parts list? Where's the genetic bang for the buck?
And the answer was in that splicing mechanism.
It was alternative splicing. Because our genes are broken up into all these different exons and introns, the cell splicing machinery has choices. It doesn't have to stitch the exons together the same way every time.
So it can skip an exon, or include one that's usually left out.
Exactly. It can mix and match the exons to create 2 or 3 or sometimes dozens of different mature mRNAs, all from a single gene.
So one gene doesn't equal one protein. It equals multiple possible proteins.
That's the leap in complexity. The source used the sentence analogy. If the gene is, "You really smell nice today," the default protein says something pleasant. But through alternative splicing, the cell can skip the nice exon, and the resulting protein's message is, "You really smell today."
Same gene, totally different meaning and function.
And a typical human gene produces 4 or 5 different versions this way. It's a huge source of our complexity. But then came the even bigger revolution, the one that violated a law of biochemistry that had stood for half a century.
The rule that all enzymes are proteins.
The bedrock rule. And this discovery, the discovery of the ribozyme, is just a fantastic story of pure, curiosity-driven science. It starts with Thomas Sick, a new professor at the University of Colorado, Boulder.
And he wasn't trying to cure cancer or upend biology. He was studying pond scum.
Literally, a single-celled organism called tetrahamina. And he chose it for a really practical reason. Its ribosomal RNA genes were prepackaged on these tiny mini chromosomes that were just super easy to isolate and work with. It was a convenient model system.
So he and his technician, a guy named Art Zog, who apparently had golden hands in the lab, were just trying to study the normal splicing process.
Right. They knew this RNA gene had a 400 base intron that had to be cut out. So they set up the reaction in a test tube. They mixed the unspliced RNA with extracts from tetrahemina which they assumed contained the protein enzymes that did the splicing. And it worked. The intron popped right out.
Standard procedure so far.
Yeah.
But then came the control experiment, the most important experiment in science.
The control. Zog ran the reaction again, but this time he left out the nuclear extract. He left out the presumed source of all the protein enzymes.
And he expected to see nothing.
Of course. No enzymes, no reaction, but he ran the gel and, astonishingly, the intron had still popped out. The RNA was splicing itself.
That must have been a moment of complete disbelief. They must have thought it was contamination. Some stray protein enzyme got into the tube.
That was the immediate assumption. So they had to prove, beyond any doubt, that the reaction was happening with RNA alone. This is where their brilliance came in.
What did they do?
They got rid of the tetrahemina system entirely to eliminate any possible contaminants. They built an artificial gene for the tetrahemina RNA sequence in bacteria. They used a single, highly purified bacterial enzyme E. coli RNA polymerase to make the RNA. Then they meticulously removed that one protein.
So the final test tube contained nothing but the pure RNA molecule, some salts, and one specific nucleotide, guanosine.
That was it. And under those pristine conditions, the intron still spliced itself out perfectly. It was undeniable. The RNA was its own enzyme.
And that's where the term came from, ribosome, a ribonucleic acid enzyme.
Sechs group had found an RNA that was an internal catalyst. At the same time, Sidney Altman at Yale found another one, RNase P, which was an external catalyst, an RNA enzyme that acted on other RNA molecules.
Together, these two discoveries just shattered the old dogma. RNA wasn't just a messenger, it was an engine, and that work deservedly got them the 1989 Nobel Prize.
It fundamentally changed the way we think about the capabilities of molecules.
So once it was clear RNA could be an enzyme, the next big question was, how? How does this floppy, charged molecule fold up into a machine? Which brings us to part 3, the architecture of RNA and the mothership.
Exactly. Function follows form. To understand how a ribozyme works, you need to see its three-dimensional structure. And this was a massive physical challenge.
It's all about charge, isn't it? Proteins have greasy bits and wiry bits so they can fold up pretty easily by hiding the grease on the inside. But RNA
is negatively charged all along its backbone. Every single phosphate group has a negative charge. So when you try to fold it up tightly to make a compact enzyme, you're forcing all those negative charges together.
It's like trying to scrunch up a ball of magnets with all the negative poles pointing inward. They just repel each other.
That's the core problem. And for a long time, it seemed insurmountable. They solved the structure of the small tRNA molecule back in 1974, that beautiful L-shaped cloverleaf. But for the next 15 years, no one could solve a larger RNA structure.
And this is the challenge that a young postdoc named Jennifer Doudna decided to take on.
Yes, the very same Jennifer Doudna of CRISPR fame. She set her sights on solving the structure of a key part of sex tetrahemina ribozyme. She had to figure out how nature solved this charge repulsion problem.
And what was the secret?
Metal ions, specifically positively charged magnesium ions. They're everywhere in the cell. Her team's structure was a breakthrough because it showed how these little magnesium ions nestle into the core of the folded RNA.
Ah, so they act like positive glue. They sit between the negative phosphates and neutralize the repulsion.
Precisely. They allow the RNA to overcome that repulsive force and collapse into a tight, stable, protein-like core, which creates the active site for catalysis. It was a fundamental principle for understanding all large RNAs.
But solving these structures with X-ray crystallography is painfully slow and difficult, and that bottleneck led to some really out-of-the-box thinking.
It really did. It led to the idea of crowdsourcing, using the wisdom of crowds to predict RNA folding.
This is the Eternity game, right?
The Eternity game. A professor at Stanford, Riju Das, had this brilliant idea to turn a horrendously complex scientific problem into a massive online puzzle game. He recruited about 37,000 players, anyone from Sudoku fans to high school students.
And their job was what? To design an RNA sequence that would fold into a specific shape?
Exactly. They were given a target shape, like a star or a cross, and they had to design an RNA sequence that, according to the rules of base pairing, should fold into that shape. The computers would provide suggestions, but often the human players, using intuition and pattern recognition, could beat the algorithms.
But how did they know if the players' designs actually worked in the real world? It's one thing for it to work in a computer model,
They tested them in the lab, they synthesized the players' winning RNA designs, and used a chemical method called shape to map their actual physical structures. And the players' designs often folded exactly as predicted.
That's amazing. And this wasn't just a game. It had a huge real-world application during the pandemic.
A massive one. In 2020, they launched the Open Vaccine Competition. They challenged the RNA players to design an mRNA sequence for the SARS-CoV-2 spike protein that would be more stable.
And stability was the #1 problem for the mRNA vaccines, right? The need for that ultra-cold storage was a huge logistical nightmare, especially for developing countries.
A complete nightmare. And the gamers came through. They designed these superfolder mRNA sequences that were up to 2 times more thermally stable than the ones designed by computers. They figured out ways to add base pairs that would make the molecule hold its shape better at higher temperatures.
So a puzzle game played by thousands of ordinary people might actually solve one of the biggest challenges in global medicine.
It's a powerful example. And now, of course, AI and machine learning are starting to accelerate this process even further, though it's still years behind protein structure prediction. We're getting closer to being able to design these complex RNA machines on demand.
And speaking of complex RNA machines, we have to talk about the biggest one of all, the mothership, the ribosome.
The ribosome, the protein factory of the cell. And for decades, it was the ultimate RNA paradox. We knew it was 2/3 RNA and only 1/3 protein.
Yet the dogma was that the protein must be doing the important work, right? The actual chemistry of linking amino acids together. The RNA was just seen as a passive scaffold.
Just a framework to hold the important protein enzymes in the right place. But one scientist, Harry Noller, a biochemist at UC Santa Cruz, just never believed it.
He was a jazz musician and a Ferrari restorer, apparently. A bit of a maverick.
And he had this deep intuition. He was convinced that RNA was the secret power source of the ribosome. His logic was that since protein synthesis is the most ancient and fundamental process in all of life, the machine that does it must be ancient. It must date back to a time before complex proteins even existed.
The RNA world.
The RNA world. Therefore, he reasoned the RNA must be the engine. But proving it required solving the structure, which was an absolutely monumental task.
This was the great crystallography race of the 1990s.
It was. Ada Yonath in Israel started the work in the 70s, famously getting the idea to use ribosomes from hibernating lizards, thinking they'd be more stable. But the final structures came out in the early 2000s from the labs of Tom Steitz, Venki Ramakrishnan, and Yonath herself.
And when Steitz's team finally got the resolution high enough to peer into the very heart of the machine, the peptidyltransferase center where the chemistry happens,
Yeah.
what did they see?
Nothing.
No protein. They saw that the active site was made exclusively of RNA. There wasn't a single protein molecule within 18 angstroms. That's a huge distance at the molecular level. It was all RNA.
The ribosome is a ribozyme.
The ribosome is a ribozyme. Harry Noller's intuition, held for decades against all dogma, was completely right. The textbooks had to be rewritten overnight. The proteins are just structural support. RNA runs the whole show. It's the strongest piece of evidence we have that life may have begun in an RNA world. Wow. From backup singer to the founding father of life itself. That's quite a promotion. Now let's bring this down from the origins of life to the limits of our own lives. Part 4, RNA in health and disease.
Right, because RNA also plays a central role in one of the most fraught topics in biology, aging. And that means we have to talk about telomerase.
The so-called fountain of youth enzyme. You see it marketed in anti-aging creams and supplements all the time.
Which is a very dangerous oversimplification.
Yeah.
Telomerase is a fascinating double-edged sword. Its role in the body is all about balance.
So what does it actually do?
It maintains the protective caps at the ends of our chromosomes. These caps are called telomeres, and they're made of short, repeating DNA sequences. Every time a normal cell divides, its telomeres get a little bit shorter.
And eventually they get too short, and the cell just stops dividing. It enters old age or senescence.
Right. That's called the Hayflick limit. But telomerase can reset that clock. It's an enzyme that adds those repeating DNA sequences back onto the ends of the telomeres, making them longer again.
And how does it know what sequence to add?
It carries its own template, and that template is a built-in piece of RNA. The enzyme is a partnership between a protein and an essential RNA guide.
So this is another ribozyme.
Not exactly. It's a reverse transcriptase that is RNA dependent. The discovery came from Liz Blackburn and Carol Greider, again, using our little friend tetrahamina. They proved RNA was essential in a really elegant way.
How did they do it?
In 1987, after they'd found the enzyme activity, they just treated their preparation with RNase, an enzyme that chews up RNA, and poof, The telomerase activity was completely destroyed. That was the proof. The RNA component was essential for it to function.
Okay, so this brings us to the double-edged sword part. On one hand, telomerase is absolutely vital for our immortal stem cells, the ones that constantly replenish our blood and tissues.
Without it, they'd die off, and we'd get diseases like aplastic anemia. But on the other hand, that exact same mechanism, cellular immortality, is a defining feature of over 90% of human cancers.
Cancers have to switch telomerase back on to become immortal and form tumors.
Exactly. So you can't just activate telomerase in your body and hope to live forever. You'd be activating countless precancerous cells. It's an incredibly delicate balance.
And we're now finding the specific mutations that flip that switch, right? The TERT promoter mutation.
Yes, this was a huge clinical advance. TERT is the gene for the protein part of the enzyme. And they found that many cancers have a single-letter typo, not in the gene itself, but in its promoter, the on-off switch right next to the gene.
And that one tiny change is enough to jam the switch in the on position.
It is. And finding that mutation in a tumor is now a major diagnostic marker. It often signals a more aggressive cancer. So RNA's role in aging is this incredibly complex regulatory dance.
So speaking of regulation, that brings us to another huge RNA story, RNA interference, or RNAi.
A discovery that also came from a humble little organism, the C. elegans worm, and again, was a complete surprise.
The researchers, Andy Fire and Craig Mello, were just trying to do a standard experiment, turn off a gene.
Right. They were using a technique called antisense RNA. The idea was to inject a single strand of RNA that would be complementary to the target mRNA, bind to it, and block it from being translated.
Simple enough. But then they ran the control experiment.
And this is where it got weird. For their control, they injected the sense strand an exact copy of the mRNA, which shouldn't do anything, but it did. It also silenced the gene. It made no sense.
They realized the active ingredient wasn't the single strands at all.
It had to be contamination with double-stranded RNA, dsRNA. So they purified the dsRNA and injected that. The result was, in their words, mind-blowing. Gene silencing was incredibly potent, 1,000 times more effective, and it was heritable. The worms passed the silenced gene down to their offspring.
That discovery won them the Nobel Prize. So what's the machinery? How does dsRNA shut down a gene?
It triggers a natural defense pathway. The cell sees the dsRNA, and an enzyme called dicer immediately chops it up into tiny little pieces called small interfering RNAs, or siRNAs.
Okay, so you've got these little RNA fragments. What happens to them?
They get loaded onto another protein called argonaute. And argonaute is the real killer. It uses one strand of the siRNA as a guide to find the matching messenger RNA in the cell.
It's like a heat-seeking missile.
It is. And when it finds its target, argonaute acts like a pair of molecular scissors and just slices the mRNA in half, completely shutting down the production of that protein.
Argonaute is the executioner, and the little siRNA is the wanted poster.
Perfect. And nature uses this system all the time with microRNAs to fine-tune gene expression. But the therapeutic potential was immediately obvious. If we could make synthetic siRNAs, we could shut down any disease-causing gene we want.
But the delivery problem rears its head again. How do you get these fragile little RNA molecules into the right cells in the body?
That was the billion-dollar question for Alnylam, the company founded on this technology. And they solved it by borrowing a trick from viruses. They packaged the siRNA inside lipid nanoparticles, or LNPs, microscopic bubbles of fat.
And they focus first on rare liver diseases, right?
Yeah. Yes, like hereditary ATTR, where the liver produces a toxic protein. Because LNPs naturally accumulate in the liver, it was the perfect target. They could deliver the siRNA, shut down the toxic gene, and they've had stunning success. It's a platform. You just change the siRNA sequence inside, and you can target thousands of different genetic diseases.
But the next frontier is much harder. Neurodegenerative diseases like ALS or Alzheimer's.
The blood-brain barrier. It's the ultimate delivery challenge. Some forms of ALS are caused by these toxic RNA repeats. The goal is to get in siRNA into the brain to chop them up. But getting LNPs across that barrier is incredibly difficult. That's the holy grail of RNA therapy right now.
It's amazing that the same molecule that can regulate our own genes is also the agent of so many of our worst enemies. Which brings us to Part V RNA in viral warfare and the vaccine revolution.
Right. RNA as the villain. RNA viruses, flu, polio, Ebola, SARS-CoV-2 are just masters of parasitic efficiency. They are stripped-down survival machines.
The very first virus ever identified, tobacco mosaic virus, was an RNA virus. And the early researchers completely missed the importance of the RNA, didn't they?
Oh, completely. Wendell Stanley, who first crystallized it in 1935, was obsessed with the protein coat. He dismissed the 6% of RNA as just some pesky contaminant. It took Heinz Frankel Conrad years later to prove definitively that the RNA was the infectious part. The RNA carried the genetic instructions.
Okay, so let's get into the strategy. How do these viruses replicate? There are two main types, right? Positive and negative strand.
Yes, and it's a key distinction. Think of it like a blueprint and a mold. A positive plus strand virus, like SARS-CoV-2, is the blueprint. The RNA genome it injects into your cell is a ready-to-read messenger RNA. Your own ribosomes grab it and immediately start making viral proteins.
So it hits the ground running.
Instantly. One of the first proteins it makes is its own RNA replicase. That enzyme then makes a complementary negative strand, which is the mold. Then it uses that mold to stamp out thousands of new positive strand blueprints to package into new viruses.
Okay, so what about the negative strand viruses, like influenza?
They're the mold. Their genome is the complementary strand, which our ribosomes can't read. So they have to package their own replicase enzyme inside the virus particle and bring it with them.
So their first step inside the cell is to use their prepackaged enzyme to make the blueprint from their mold.
Exactly. They have to make the positive strand first, and then the replication can take off. It's one extra step.
Now, the key to why these viruses, especially RNA viruses, evolve so fast is that their replicase enzyme is sloppy.
Extremely sloppy. It's very error-prone. It makes a mistake roughly once every 10,000 bases it copies. A lot of viral genomes are about that long, which means basically every new virus that's made has a typo in it somewhere.
It's mutation on hyperdrive.
It is, and this was demonstrated in this incredible experiment by Saul Spiegelman back in the 60s. He took a phage and forced it to replicate faster and faster in a test tube. He created an evolutionary pressure cooker.
And what happened to the virus's genome?
It got shorter and shorter. The replicase started making mistakes and skipping huge chunks of the genome because it was faster. The original 3,300 base genome was whittled down to a 300 base little monster that had lost all its genes except the tiny bit needed for the replicase to bind to it. It was a pure replication machine.
That's Darwinian evolution in a test tube. And it perfectly explains how we get variants like Omicron with 35 mutations in the spike protein. It's not planning, it's just making millions of random typos, and the ones that help it survive and spread are the ones that take over.
Precisely. But this villain molecule, this agent of destruction, became the hero of the pandemic through the mRNA vaccine revolution.
An idea that started way back in the late 80s with a grad student named Bob Malone.
Malone had the key insight about safety. Gene therapy at the time was focused on DNA, but there's a huge risk. DNA can permanently integrate into your own genome, potentially causing cancer. It was a terrifying risk that sadly did happen years later.
But mRNA is different.
It's transient, it can't get into the nucleus, it can't integrate into your DNA, it delivers its instructions to the cytoplasm, the cell makes the protein for a little while, and then the mRNA just degrades and disappears. It's a much safer platform.
So to make this idea a reality, 3 huge puzzle pieces developed over 30 years had to come together. Puzzle piece 1, making the mRNA.
This came from Bill Studier's basic research on a bacteriophage called T7. He discovered that the T7 virus has its own RNA polymerase that is an absolute beast, a hyper-efficient RNA-making machine.
It's very specific, right?
Very. It only recognizes one specific 17 base pair starting signal. So you can take any gene you want, like the spike protein gene tag, that T7 starting signal onto the front, add the T7 polymerase, and it will churn out mountains of pure mRNA. It made mass production feasible.
Okay, puzzle piece 2. The delivery vehicle, you still have to get it into the cell.
The LNPs, again, this goes back to Phil Fellner's early work. And the incredible story is that his boss at the pharma company shut down the LNP project in 1988 for not being profitable enough, but said to him, "I bet this will be really important around the year 2020.
You cannot make that up.
You can't. The technology was later refined by people like Peter Cullis, who developed these brilliant charge-switchable LNPs. They're neutral in the bloodstream, so they're stealthy, but they become positively charged inside the cell, which helps them rupture and release the mRNA.
And then puzzle piece 3, the absolute key to making it work in humans, hiding the mRNA from our own immune system.
This is the incredible story of Catalin Carrico and Drew Weissman at the University of Pennsylvania. Carrico was a brilliant, dedicated RNA scientist who was repeatedly denied funding and demoted because her work wasn't seen as mainstream.
And she met Weissman, an immunologist, at a Xerox machine.
A legendary meeting. They realized that our cells have alarm systems that are exquisitely tuned to detect foreign RNA and trigger a massive inflammatory response. They had to figure out how to cloak the mRNA, to sneak it past those alarms.
And the secret was a tiny chemical tweak.
A tiny tweak. They found that if they swapped out one of the 4 RNA bases, uridine, for a slightly modified version called pseudo-uridine, or pseudo-U, the immune sensors were basically blinded. They didn't recognize it as foreign anymore.
The cloaked mRNA could now hang around long enough to produce a ton of spike protein and properly train the adaptive immune system without causing a dangerous inflammatory storm.
That was the final critical piece. In 2020, BioNTech and Moderna put it all together. The T7 production, the LNP delivery, the pseudo-U cloaking, and achieved those stunning 95% efficacy rates. It looked like an overnight success, but it was built on 30 years of grit and basic research.
And now that platform is being aimed at everything from RSV and the common cold to personalized cancer vaccines and protein replacement therapies. It's a new era of medicine.
It truly is. And that brings us to the final and perhaps most powerful role for RNA. Part 6. The ultimate catalyst RNA-guided genome editing.
CRISPR. This is where RNA isn't just carrying a message, it's providing the architectural plans to rewrite the source code of life itself.
And just like so many of these other stories, it starts with a battle between bacteria and viruses. CRISPR is a natural bacterial immune system.
Right, it's how bacteria remember and fight off viruses that have attacked them before. It relies on 2 components, the Cas9 protein,
which is the molecular scissors,
and a guide RNA,
which is the GPS. The breakthrough from Jennifer Doudna and Emmanuel Charpentier in 2012 was realizing you could reprogram that guide RNA.
You can give it any address you want.
Any address in the genome. The guide RNA uses simple base pairing to drag the Cas9 scissors to that precise location and tell it where to cut the DNA double helix. It democratized gene editing overnight.
But the real editing happens after the cut, right? When the cell tries to repair the damage.
Exactly. And scientists can hijack 2 different natural repair pathways. The first is the fast and sloppy one.
Non-homologous end joining, NHEJ.
The cell's emergency response. It just jams the broken ends of the DNA back together, and in the process, it often inserts or deletes a few bases. This causes a frameshift mutation, which usually just inactivates the gene.
So if you want to turn off a bad gene that's making a toxic protein, this is your tool. Cut lets a cell mess it up, and the problem gene is silenced.
Precisely. But if you want to do something more precise, like correct a mutation, you need the second pathway.
Homologous recombination, the copy and paste function.
For this one, you not only cut the DNA, but you also provide the cell with a donor template, a correct piece of DNA sequence. The cell then uses that template as a guide to repair the break perfectly, pasting in the correct sequence.
This is how you could, in theory, correct the single-letter typo that causes sickle cell disease.
And we are already seeing this move into the clinic. The first approved CRISPR therapy, exa-cel, uses the turn-it-off approach to treat sickle cell by reactivating a dormant fetal hemoglobin gene. It's proof of concept that this RNA-guided medicine is real.
And the tools are getting even more precise. Now we have dead Cas9.
A brilliant innovation. This is a version of Cas9 that has been neutered. It can still use the guide RNA to find its target DNA address, but its blades are dull. It can't cut.
That's the point.
You can fuse other tools to it. You can attach a gene activator to turn a gene on, or a repressor to turn it off, or, most excitingly, you can attach a base editor.
Which can do what?
It can perform chemical surgery on a single DNA letter. It can go to a precise spot and, without cutting the DNA at all, chemically convert a C to a T or an A to a G. It's like a find and replace function for the genome with much lower risk of off-target errors.
This power is just. It's almost unimaginable, which of course raises huge ethical questions.
Yeah, massive ones. The power to make permanent, heritable changes to the human germline is now a reality, and it requires us as a society to draw some very clear lines about what is and is not acceptable.
And as we close, we have to acknowledge the great unknown. The final, unfinished chapter of the RNA story, the so-called dark matter of the genome.
Long non-coding RNAs, or LNC RNAs. We now know that our cells transcribe hundreds of thousands of these mysterious RNA molecules from the vast non-protein coding regions of our DNA. And for the most part, we have no idea what they do.
But if the history of RNA has taught us anything,
it's that we dismiss junk at our peril. Ribozymes were curiosity, microRNAs were noise, telomerase RNA was a fluke. It seems almost certain that these LNC RNAs are playing vital regulatory roles that we are only just beginning to uncover. They are the next frontier.
It's an incredible arc to synthesize. RNA starts as a passive link, then it's a catalyst, then a regulator, and now it's the engine behind our most advanced medical technologies.
And through it all, there's that recurring theme. RNA provides the guide through its sequence and its structure, and a protein often provides the catalytic act. That partnership is the foundation of modern life.
And when you step back and look at where these breakthroughs came from, it's really striking.
It is. Ribozymes and telomerase came from pond scum. RNA interference came from a tiny worm. CRISPR came from bacteria.
Every single one of these world-changing revolutions, the ones that are now curing human diseases, came from basic, curiosity-driven research. Scientists who were just trying to figure out how a weird little organism worked.
And yet we see this trend, this pressure to only fund research that has a clear short-term path to a commercial product or a cure. Funding for the pond scum research is harder and harder to get.
And that brings us to our final thought for you to mull over. Given that the truly transformative leaps in medicine have almost always come from these unexpected corners of biology, from scientists driven by pure curiosity, what unexpected, lowly critter out there might be holding the secret to the next great medical revolution?
A secret that might just be waiting for someone curious enough to look. The biggest answers are rarely where we expect to find them.
Thank you for joining us for this deep dive into the world of RNA. We'll see you next time.